All right, well, good morning. Welcome to our second Genomic Epi Seminar of the season. I just wanted to remind everybody that the format for this call has changed a little bit in that, this meeting is primarily invite only right now. It is intended for field epi regional staff, DSI Central Office Program Area Leads. We also have members from DDP, the HAI team, the HIV and hepatitis group, environmental health, as well as our DCLS laboratorians joining us for this call now. And we do have some overarching, can't talk, goals and aims, but I did just want to mention that we do intend to have kind of a blended mix format. Last week was more of a straight PowerPoint presentation, and today is more going to be of a training style format with a guest speaker. And in the future, we plan to do some journal club style meetings, but we will do our best to keep these going at least once a month up until the holidays, and attempt to mix up the presentation materials to try to address needs that go across all those different attendee areas. So without further ado, I wanted to introduce our guest speaker today. Dr. Rebecca Wattam is a research associate professor. She works at the University of Virginia Biocomplexity Institute. Her background heavily focuses on infectious diseases. She received her PhD in veterinarian science and entomology from the University of Wisconsin-Madison in 1992. She has a wide variety of interests, including bacterial pathogens, and focuses on comparative genomics. So with that brief introduction, I will turn it over to Rebecca for the presentation part of this seminar today. And she did say that she is okay with questions if you want to drop those in the chat as we go along. I can help monitor that and bring those up. Or if you want to raise your hand with the raise hand emoji, we can try and address those as Rebecca has pauses throughout the training. All right, I will turn it over to you, Rebecca.
Thank you, Jenny, and thank you all for inviting me. I was present for last month's talk by Logan, which I thought was excellent. Today is going to be somewhat of a follow-on to that. We're going to talk a lot about phylogeny and using it for pathogen identification. One thing I want to point out straight up from the top is that I'm a biologist. I am not a bioinformatician, and I'm not a computer scientist. Yet sometimes I'm able to masquerade as one because I know where there are online sources that I can do my analyses at. And so I'm hoping today to show you some of those and help you figure out how you could generate a phylogenetic tree if you so desired to do so. So I have to keep reminding myself, because generally to me, phylogenetic trees are the end, the thing I'm searching for, but it's just a hypothesis. And it's a hypothesis based on the available data. It's not written in blood, it's not written in stone, and sometimes if you add more data, the tree will change. But basically what it is a visual representation of how these organisms are related and how they are changing over evolutionary time. And this, I know Logan had a drawing of it last month, but this is out of Charles Darwin's notebook, and I think it's one of our very first renderings of a phylogenetic tree. So Logan talked about tree anatomy, and I just want to refresh that in your mind briefly. Some trees have a root, which I'm afraid to use my mouse because sometimes it goes forward. But here it is. Some trees have roots. Some trees, or all trees, have nodes. And the node is really, you're assuming that there was some hypothetical or ancestor. And at this point in history, some of his kids became lizards and some became these other guys. Whoops. They're branches. Now, branches may or may not be indicative of the amount of change depending upon if you have a scale. If there's a scale present, and that's generally when you have something like sequence data, the length of the branch can indicate change between the organisms. And obviously, I mean, I created this myself. This isn't a true representation of what's happening in the evolution of vertebrates. And then the leaves or the tips are the organisms, but they can also be the genes or the proteins. So rooting is important because depending upon if a tree is rooted or not, it is going to influence your interpretation of a tree. A tree can be unrooted, like you see up here. It can be midpoint rooted. Now, when it's midpoint rooted, you're assuming that the organisms or genes or whatever it is that you're comparing, that they're evolving at a constant rate or at the same rate. And when you're looking at things like within the same genus or species of, well, I work a lot with bacteria, so I'm always thinking about bacteria. That's a sometimes safe assumption. If you're looking more broadly, sometimes that's not a safe assumption because some things are evolving much more rapidly than other things. And then we have outgroup rooting. And outgroup rooting is where the person who generated the tree is making an assumption and saying, this is the ancestor, and I'm looking at everything, how that relates when this is my ancestor. And in this example, we have the frog, or the amphibian is the ancestor to the reptiles and the mammals. So what are these trees based on? Well, they can be based on physical characters, and they can be based on sequence characters. I really like this rendition of the evolution of the eye, starting with sponges who are eyeless. And then it's showing you that each of these organisms have a complex like eye structure. And so you may be thinking, my goodness, how can you tie these things together? They don't even look the same way. They don't even look the same. Like between the jellyfish and the insect, things are really different. The octopus and the vertebrates look somewhat similar. So it can't be just on overall morphology looking at that and saying, well, those are more closely related because the octopus eye looks a lot more like the vertebrate eye. Well, those things are based on physical characters, and generally when a tree is built, it's creating a data matrix of whatever characters you're using. And so in this instance, we have our organisms across the top and in the columns, and our the things that we're ranking or scoring are the rows. And you can see that some of them have a 0 or a 1. And this one with the physical characters, the 0 indicates that it's not present, and the 1 indicates that it is present. And so the bioinformatic pipeline that's going to create a tree based on this is going to go through and rank all those and then generate the tree based on those things. So it's not what it looks like. In this case, it's the presence of a particular organelle that it's looking at. So characters can also be sequence data, and I think looking at this, which is a multiple sequence alignment of the nucleotides of a particular gene. When you look at this, you realize right away that when you're looking at physical characters, those are generally numbering in the tens to base the tree. Once you get into proteins, if you're doing protein sequence, it could be the hundreds, maybe the thousands. But when you get into genes, you're talking about the thousands to maybe some genes are really long, multiple thousands. And when you're talking about whole genomes, you can have hundreds of thousands, and in some cases, maybe a million characters to base these trees on. So this is just an example where it's oriented a little bit differently. I have, and I just sort of made this up, but I have the organisms now as the rows, and the columns are the sequence at that particular position in an alignment. And so you can tell that we are already getting a little more complicated. It's not just 0 or 1. You have 4 choices of things, the C, the G, the A, or the T, that can be used in the alignment. And just think about this as when you have hundreds of thousands of columns of data.